Variation in traits within a population can be explained in part by heredity, the passing of traits from one generation to another. Sexual reproduction allows for genetic traits to become combined in a seemingly unlimited number of combinations. However, there are specific rules that govern how these traits are inherited. These rules were discovered by Gregor Mendel, an Austrian monk who, through his experiments with pea plants, became known as the father of modern genetics. By analyzing his experimental findings, we can come to understand and apply his model of heredity. Mendel probably owes much of his success to his choice of experimental subject, the pea plant. Pea plants are exceptionally easy subjects to study in a genetic experiment due to their structures and functions. Pollen, or the sperm, of plants is housed in the stamen of flowers. The ovum or eggs are found deep within the carpel. This carpel develops into the seed-containing fruit of the plant. Each seed, or pea, is an individual organism that can develop into individual pea plants. So Mendel could grow a lot of peas from one genetic cross. In each of his crosses, Mendel used a paintbrush to transfer pollen from one plant to another. That way he could control the fertilization between parental generations, cultivate the many offspring that resulted from the cross, and observe the traits. In the mid-1800s, when Mendel was doing his experiments, most biologists subscribed to the idea of blending inheritance, a general model in which inheritance involved the blending of parental characteristics in the offspring. This model fit well with some observations of human inheritance. For example, you often look a bit like both of your parents. But it did not explain the results of Mendel's experiments with pea plants. Take a look at these examples. When he crossed a tall and a short pea plant, all of the offspring were tall, not medium. And then when he did self-fertilization with those tall offspring, he noticed that about 3/4 of the offspring were tall, while 1/4 of the offspring were short. Based on the observations of 7 different distinct traits in pea plants, Mendel was able to come to the following conclusions. First, Mendel determined that different versions of genes existed and therefore accounted for variation in a specific trait. These different versions of genes are called alleles. Take flower color, for example. In pea plants, flowers could be purple or white. We now know that flowers are purple because the gene codes for an enzyme that synthesizes purple pigments. That same gene in a white flower contains a sequence of bases that does not code for the enzyme. Therefore, no pigment can be produced, and the flower appears white. Mendel also noted that each individual has 2 copies of each gene, one inherited from each parent. It is the interaction between these genes that will ultimately determine the physical traits of each individual. Next, for each of the 7 traits Mendel studied, he noticed that one trait seemed to mask the other. When crossing a true breeding plant with green pods and one with yellow pods, he observed that 100% of the offspring had green pods. Therefore, the green trait is dominant over the yellow trait. Self-pollination of that F1 generation plants resulted in a 3-to-1 ratio between green and yellow pods. There's only one way that a pea plant or any organism could show the recessive trait. That's if they have 2 recessive alleles. Using this information, we can characterize the organism's genes or genotype using letters. Capital letters denote the dominant allele, while lowercase letters represent the recessive allele. The genotype of an organism determines their appearance, or phenotype. Notice that if an organism has at least one dominant allele, they will have the dominant phenotype, or green in this case. If they have 2 recessive alleles, they'll have the recessive trait, or yellow pea pods in this case. We use several terms to describe the organism's genotype. First, homozygous dominant refers to an organism that has 2 copies of the dominant gene. Homozygous means same. Homozygous recessive means the same thing, 2 of the same alleles, but these 2 alleles are recessive. Finally, heterozygous describes a genotype of an organism that has 1 dominant allele and 1 recessive allele. Remember, each organism has 2 copies of each gene, 1 from each parent. But when an organism produces gametes, these alleles separate from each other. This is known as the law of segregation. The separation of these alleles can be easily observed through the steps of meiosis. Notice an egg or sperm will only receive one allele or the other. When studying multiple traits at once, Mendel noticed that the inheritance of one trait did not influence the inheritance of another. This is because alleles of two or more genes separate independently when packaged into gametes, since they are found on separate chromosomes. Notice we can get a green allele paired with a purple, a green with a white, a yellow with a purple, or a yellow with a white. This law of independent assortment further explains how variation within offspring of the same parents exists. We can use Mendel's discoveries in a variety of ways. First, test crosses can be used to determine the genotypes of the parents. Take this example of a cross between a purple-flowered plant and one with white flowers. We know that the white flower plant has to be homozygous recessive, 2 little p's. But the purple plant could have a dominant gene with another dominant gene, homozygous dominant, or it could be heterozygous with a dominant and a recessive. So we can do a couple predictions. Here, if we used the fact that the purple plant was homozygous dominant, we would see that all of the offspring would be purple. If we use that same prediction with a purple plant that was heterozygous, we would see that only 50% of the offspring would be purple. So when we actually do this cross and we count all of the offspring, if all of the offspring are purple, we can assume that the parent was homozygous dominant. However, if only 50% of the offspring are purple, then we can assume that the parent has a heterozygous genotype. We can also use Punnett squares to be able to predict the traits of an offspring of a genetic cross using Mendel's model of inheritance. Punnett squares are a tool that we can use to visualize how the alleles are packaged into gametes and then are combined during fertilization. We then have to apply the rules of dominance and recessive alleles to interpret the phenotypic outcomes of the offspring. So take this cross, for example. We can have peas that are round or peas that are wrinkly. The round trait is dominant over the wrinkly trait. So if I take a pea plant that is heterozygous for the round trait and complete the cross, I can see that 3 of the squares show a round phenotype and 1 shows a wrinkled phenotype. Therefore, the expected phenotypic ratio in the offspring is 3:1. We can use larger Punnett squares to predict the outcome of dihybrid crosses, one that involves 2 traits. So take a look at this example. Let's say I'm focusing in on yellow seeds, plants that are homozygous or heterozygous for yellow seeds and heterozygous for round seeds. Now, what we'll have to do is figure out what are all the possible combinations that could organize within the gametes. Well, we could have a dominant yellow gene and a dominant round gene. Dominant yellow, recessive wrinkled, recessive green, dominant round, recessive green, and recessive wrinkled. The same would happen in the other parent. Then when we fill in the table, we get all of these different possible genotypes, and then we have to go through and figure out, well, how many have each phenotype? So 9 of them will have the phenotype yellow and smooth. 3 of them will be yellow and wrinkly, 3 of them will be green and smooth, and 1 of them will be green and wrinkly. So we can see this 9:3 to 3:1 ratio in the phenotypes. There is a much easier way to make this prediction than using one of those giant Punnett squares. And what we can do is just focus on one trait at a time, because remember, the law of independent assortment says that one trait's inheritance doesn't affect the others. So we can make the prediction of just the yellow trait and then just the round trait, and then we can take those probabilities and multiply them together. So 3 yellow seeds times 3 round seeds is 9. Green and round, 3 times 1 would be 3. Same thing here, 3 times 1 is 3, and green and wrinkled, there would only be 1 of those. So we still get that 9:3 to 3:1 ratio, and it becomes much easier than trying to fill out one of those giant Punnett squares. Mendel's model of inheritance was groundbreaking and is still used today as a model for the inheritance of traits. However, not all traits are controlled by simple dominant recessive alleles. In fact, the inheritance of most traits goes well beyond Mendelian genetics. So stay tuned for the next podcast to learn about the other modes of inheritance. In the meantime, I hope you learned a little bit from this podcast about Mendelian genetics. Thanks.